Restriction maps and restriction enzymes. Restriction enzymes or restriction endonucleases are enzymes which cuts DNA at or near specific recognition nucleotide sequences known as restriction sites. These restriction enzymes recognizes a specific sequence of nucleotides and produce a double-stranded cut in the DNA. For example, the popular enzymes EcoR1 and HIN-D3 are widely used in labs. EcoR1 cuts where it finds GAATTC between the G and A. Actually, it cuts both complementary strands, leaving an overhang on each end. HIN-D3 cuts at AAGCTT and cuts between the A's. These sticky ends of a few bases in single strands make it possible for the fragments to reform, making possible the insertion of DNA into vectors for cloning and sequencing. Some enzymes cut in the middle and result in blunt ends with no overhang. These are given in the figure. Restriction map is a map of non-restriction sites within a sequence of DNA. Computing restriction maps is a common and practical bioinformatics calculation in the laboratory. Restriction maps are computed to plan experiments to find the best way to cut DNA to insert a gene to make a site-specific mutation or for several other applications of recombinant DNA techniques. By computing, the laboratory scientists can save the trial and error at the laboratory. Keeping this in mind, we are going to write a program that will look for restriction enzymes in a sequence of DNA and report back with a restriction map of exactly where in the DNA the restriction enzyme appear. Planning the program. Here we need to use the technique learned earlier to find the motifs in sequences with Perl using regular expressions. So now we are going to write a program that includes restriction enzyme data translated into regular expressions stored as values of the keys of the restriction enzyme names. DNA sequence data will be used from the file and the user will be prompted for names of restriction enzymes. The appropriated regular expression will be retrieved from the hash and we can search for all instances of that regular expression and their locations. Finally, the list of locations found will be returned. Restriction enzyme data. The restriction enzyme data is available in a variety of formats. Rebase is a database of information about restriction enzymes and DNA methyltransferase. Rebase contains an extensive set of references, sites of recognition and cleavage, sequences and structures. It is one of the longest running biological databases. The restriction enzyme database will look something like the table given on the screen. The table contains 3 columns. The first one is the name of the enzyme, the second is recognition sequence and the last one contains other enzymes capable of cutting these sequences. Our first task is to read the file and get the names and recognition site or recognition sites for each enzyme. To simplify, we discard the parenthesized enzyme names. The procedure is discard header lines and for each data line remove parenthesized names for simplicity's sake, get and store the name and the recognition site. Translate the recognition sites to regular expressions but keep the recognition site for printing out results. Then return the names, recognition sites and the regular expressions. The pseudocode to discard the header lines is for each line if the regular expression Rich Roberts break out of the for each loop. Now we can further expand the pseudocode to do the task specified. First one is discard header lines. This keeps reading lines up to a line containing Rich Roberts. The code is for each line if Rich Roberts break out of the for each loop. Then for each data line, split the 2 or 3 if there is a parenthesized name fields 2 fields equal to split with space. Then get and store the name and the recognition site. The code will look like $name equal to shift array fields and $site equal to pop array fields. Then we have to translate the recognition sites to regular expressions, but keep the recognition site for printing out results. Then return the names, recognition sites and the regular expressions. First we want to extract the name and recognition site data from a string. The most common way to separate words in a line of Perl, especially when the string is nicely formatted, is with the Perl built-in function split. If there are 2 or 3 fields per line that have white spaces and are separated from each other by white space, they can be get into an array with the following call to split. Here the lines are assumed to be stored in a special variable @_. The statement $name, $site equal to split and space as an argument. The @field array may have 2 or 3 elements depending on whether there was a parenthesized alternate enzyme named, but it is necessary to know the first and the last elements. $name equal to shift fields and $site equal to pop fields. Now the problem is to translate the recognition site to a regular expression. By the sample recognition on Rebase, it can be noted that the cut site is represented by the caret symbol. This does not help make a regular expression that finds the site in sequence. So we have to remove it. Also notice that the bases given in the recognition sites are not just the bases A, C, G and T, but they also use the more extended alphabet as presented in the table. These additional letters include a letter for every possible group of 2, 3 or 4 bases. They are really like abbreviations for character classes in that respect. So we have to write a subroutine that substitutes character classes for these codes. Of course, Rebase uses them because a given restriction enzyme might well match a few different recognition sites. The following program is a subroutine that, given a string, translates these code into character classes. IOB to regular expression is a subroutine that, given a sequence with IUB ambiguity code, outputs a translation with IUB codes changes to regular expressions. These are the IUB ambiguity codes. R equals G or A, Y equals C or T, M equals A or C, K equals G or T, S equals G or C, W equals A or T, B equals not A, C or G or T, D equals not C, A or G or T, H equals not G, A or C or T, B equals not T, A or C or G, and N equals A or C or G or T. The subroutine IUB to regular expression will look like: first we have to assign the array to my $IUB. Then the regular expression variable is initiated and the IUB to character class is initialized with the letters A, C, G, T, etc. Then remove the character signs from the recognition sites. The statement will look like $IUB equals tilde S within slashes character symbol then /G. Translate each character in the IUB sequence. A for loop is used for this. The loop variable I is initialized to 0 and it is tested up to the length of IUB variable. Then the regular expression is concatenated with the IUB to character class and the substring $IUB $I1. Then return the regular expression. But we have to plan what data we want in return. We can plan the program to return 3 data items per line of the original Rebase file: the enzyme name, the recognition site and the regular expression. For this, we can use an array that stores these 3 data items in 3 consecutive slots. When we want to read the data, we have to read groups of 3 items from the array. It is possible, but the lookup is a bit difficult. Using advanced Perl, we can create our own complex data structures. Using the split mentioned above, we can have a hash in which the key is the enzyme name and the value is a string with the recognition site and the regular expression separated by white space. Then data lookup can be made fast and just extract the desired values using split. The given example program shows this method. Here is a subroutine to pass a rebase data file. It is given the name pass rebase, which is a subroutine to return a hash where key equals restriction enzyme name and the value equals white space separated recognition site and regular expression. The subroutine will go like this. Sub pass rebase, my $rebase file is initialized, then the variables rebase file and rebase hash is initialized, and name, site and regular expressions are the other variables. Then we are reading the rebase file. Rebase file equals get file data of rebase file, where get file data is a subroutine we have used earlier. For each rebase file, discard header lines. For this we are using the regular expression and next. Then discard the blank lines. Split the 2 or 3 if includes parenthesized names fields. My fields equal to split called with the space and the array. Get and store the name and the recognition site. Then remove parenthesized names, and for simplicity we are saving the first and last only. The name equal to shift fields and site equals pop fields. Then translate the recognition sites to regular expressions using the subroutine IOB to regular expression, which is called with the variable site. It is returned to the variable regular expression. Store the data into the hash. Rebase hash of $name equal to site and regular expression. Return the hash containing the reformatted rebase data and return rebase hash. This pass rebase subroutine does a lot of things. Now we will see how these things are done in this subroutine.